All right, so I'm going to talk to you about learning from unpopular activities. And now that I removed the second part of the title, I realize it doesn't sound as cool. But what we're going to do is focus on unknown functionality and how important that is for understanding the functionality of organisms. So I started thinking about protein function in 2001 when I joined this particular lab. Every person on that picture had thought about protein function. And those of you who know who these people are, so there is Yana Bromberg and Raj Nair and Jinfeng Liu and Darish Perbilsky. So all of these people still think about protein function. So this is saying something. One of the people that you may have heard talking here is Dr. Burkhard Rost. He was the PI there. He's there, so I'm not sure I'm allowed to say where. So if you can figure it out. All right. So the reason I bring this up is because I came to the group thinking about protein function, thinking that this is something really well defined, but apparently it's not. And wasn't then, I don't think it still is. So when I think about protein function, I think about this notion, this is a quote from 2003 paper from them, where it says, here we use the generalized operational notion that function is everything that happens to or through a protein. Right? So if you disagree with my definition of function for this talk, I'm really happy to chat about this afterwards. But for now, we're going to talk about that. And then I'm going to sort of narrow it down a little bit, but I'll tell you when I do. So why is function so difficult, function prediction so difficult? One of the reasons is this. So I stole this data from a paper on the roadmap of function prediction. So this is from a meeting that happened last year about trying to figure out what we need to do about function prediction. This is a figure representation of the numbers of Go annotations that we have for every particular organism, right? So for example, the human takes up 20% of that chart. So 20% of Go annotations are human annotations, right? And then we have mouse and Apheliana and rat, and so all these other wonderful model organisms that are up there. So E. coli, which is actually also really well annotated, is only about 2% of Go data, right? So if you are interested in microbes, which I am, tough luck, right? So there really isn't very much experimental annotation for microbial organisms out there. And others make up 14%. So every other organism you can think of is in that 14% of Go annotations, right? Okay, so that's one problem. Experimental annotations are very unevenly distributed. I used this slide for many years because I still find that absolutely amazing. There are plenty of misannotations. So in 2009, Alex Schnoz in Patsy Babbitt's group had actually evaluated how much misannotation is there across databases. And what she found is that it increases with time. So this is the problem of transfer of function annotation by homology. So we started out okay in the 90s with no annotations, but also the ones we had were actually good. And then we went on to in 2005, which is where this particular image ends, and 40% of the functional annotations in databases were misannotated. And this 40% is not evenly distributed. So if you have a particular family you're really interested in, that one can be 80% misannotated, right? Depends, right? So this is another problem. And then even the predictions are not so easy to come by. So if you go, as I did a couple of days ago, to Uniprot website and ask the question, what do we have in terms of annotations? There is 560,000 Swissprot manually curated annotations in Uniprot. And then there is the number that I can't even imagine, 248 million proteins, right? That was not there before. I don't know when it got there, but 248 million proteins that we have in Uniprot. And then we ask, okay, which ones are annotated? So for Swissprot, actually 81% are annotated, right? Experimentally annotated. But for Tramble, only 14% are. right? So if you're just starting out, if you're a graduate student or a postdoc just starting out on function, hey, there is all that you can annotate. Just go for it, right? But this is how we live, right? We've got limited annotations. And then over time, you realize that everything that you would like to evaluate and study, someone had tried before. So here's a 2017 Simon Kassif paper where he actually relates the number of protein family members to the number of publications on the topic. So the more protein family members there are, the more studies there are. And that's why there is 1 million studies out there on the functionality of BRCA1, BRCA2 genes, right? And probably not that many on your favorite bacterial protein, right? So this is the state of events that we're in. And so for the rest of this talk, I'm going to use these couple of rules for protein function prediction, right? So one is any one protein can have many functional labels. Depending on what your annotation is, you could have as many functional labels as you want. And individual tools will only predict some of those functional labels. And at any given point in time, you may or may not have all functional labels that you are interested in for one particular protein. And very importantly, some predictions are possible ab initio. So basically, if you don't have a reference, you can make some predictions for those functions. So for instance, subcellular localization via signaling is one of those things that you can do with ab initio, technically, ab initio for the protein sequence. And others require some sort of comparison. And up until the recent explosion of this deep learning story, the comparisons were homology. So all you could do is compare a new protein to an existing protein with annotations, and that's how you would do function transfer. Okay, so that last part, as I said, is actually getting some sort of input, and I don't know yet what kind of input, but from deep learning. And the idea is that now all of a sudden you don't need to necessarily use existing annotations, use proteins of existing annotations in order to predict the function of a particular sequence, assuming that these deep learning models actually learn the language of life, right? So if they do, they can figure out what a particular protein means without having necessarily a specific comparison. Maybe not, right? So let's talk a little bit about this. Can we improve protein function annotation? So those of you who know the Pokeball up there, we have the space of protein sequence, right? So all the protein sequence that exists in the world, regardless of whether we know it or not, all protein sequence that exists in the world. And we've got some of it that has protein functions that we do not yet know about, and some of it that has protein functions that we can annotate, right? So this is talking about mostly the ontologies that we currently have, right? So some functions, I believe, truly believe, we don't know everything. So some functions we don't yet have annotations of, molecular functions. When we think about the proteins that we know, this, and please don't quote me on the scales. I don't actually know how many proteins we know in terms of everything that's out there. But let's say there is some known proteins over there. And these known proteins are actually falling into the space of known and unknown, right? And why do I say this? Well, if I'm going to run ahead of myself and tell you, we looked at individual organisms, at microbes, and for every particular single microbe, we know that there is an average 100 proteins that have no homologue anywhere else. And I can guarantee you that none of them have annotations, right? Okay, I can't guarantee you that. Maybe one of them has annotations, but otherwise, it's really difficult to explore that space because they have no homologues, right? Okay, so known proteins are split into unknown world and known world. And then there are these proteins that are outside of that known protein space. They exist somewhere in this world, we just haven't seen them yet. And the best repository, best resource for these proteins is microbiomes, right? So when you go to sequence the metagenomic data of I don't know, deep sea oceans, right? So people who are looking at Tara Oceans expeditions, or the mid-Atlantic, mid-Pacific ridge. I'm sort of losing my touch with geography, but there's all of these wonderful expeditions out there where people get microbiota and they try to annotate it. And these split into unknown unknowns and known unknowns. I'm sorry for citing Rumsfeld, but the idea here is that proteins which we haven't seen could be of unknown functionality as well, right? Okay, and so here's the fun part of this. This little piece is where this little circle in the middle is the annotated proteins, whether by homology or because someone actually did the experiment. And where most of the protein prediction methods focus is that space over there, right? So we're going to predict the proteins that we know exist, and we're going to try to annotate the function that we know exists, right? So if we're interested in trying to figure out unknown unknowns, how do we do this? So those of you who were in the keynote earlier, this is Burkhardt talking about trying to annotate function by homology, and structure by homology. And in that particular space, this is basically just structure. So people think that structure has a relationship with function, right? And it does. It has a relationship with function, but it's not a one-to-one mapping. So in 2002, Burkhardt had created that same version of that same plot for function annotation. A postdoc in my lab, Yannick Malic, whom I'm going to show on the next picture for some reason, he didn't make it to this slide, but Yannick had actually created a new version of this together with Martin Steinegger to use MM6 data, right? So rather than running BLAST, which takes forever, we ran MM6 on all proteins that had enzymatic annotations, so EC number, enzyme commission number. And we try to differentiate proteins that have alignment of proteins that have the same EC number from that of proteins that have a different EC number. And what you see there is that blue line separates it fairly well. The black line was the original HSSP line. So this is basically just a sort of re-optimization of HSSP into HFSP for using faster tools and better ways of addressing the existing data sets, right? So Yannick built that method, and if we ask how well does this method perform, there's Yannick. If we ask how well this method performs, we see that at this zero cutoff, we have 92% precision, more or less, depending on which date of the data set you have in your hands, and about 53 to 64% recall, right? So we can identify functionality by homology for these proteins, for 50% of these proteins, more or less, right? Very precisely. So Yannick said, okay, well, we don't have annotations, EC annotations for a large number of proteins, right? For some of them, we don't. So how do we annotate function in a way where we don't have EC number? And again, I remind you that microbes are the ones that have the largest number of yet unannotated proteins. So that is what he looked at. So what he looked at was this set of 9,000 microbial organisms, fully sequenced microbial organisms, for which proteins were predicted. And he said, I'm going to use this method, this HFSP method, to try to pairwise align the proteins in this data set. And we're talking about 37 million proteins. So it was actually quite nice that we had a fast running method for doing this, because 37 million by 37 million is a really long time, right? So when he did the alignment, we then computed, so he did the MM6 alignment, we then computed the HFSP scores, and we then applied clustering techniques to this particular set of proteins. This took 2 years. Just saying that it's not an easy thing to do, even though it sounds very, very easy, right? To theoretically. So logically, it sounds easy. Methodologically, it's impossible, or possible, but after 2 years. So what he did is he clustered these proteins into what I will continue to call from now on fusion functions. The database is called FusionDB. The clusters, functional clusters of these proteins are fusion functions. And so, of course, it took us this long to get there. And the next question was, so if I get a new protein, how do I know which fusion functions it falls into? Right? Do I have to rerun everything again? It's terrible. And so we asked a different question. We said, how do we assign a particular protein to these fusion functions? Now, remember, these are fusion functions that have IDs and no names, right? This is an important point. These are unknown functions. Okay, so what we did is trained a Siamese neural network. And if you're interested in the detail, I'm happy to talk to you about how we did this. But we trained the Siamese neural network to recognize two proteins as coming from the same fusion functions. Now, we looked at gene sequences rather than protein sequences. And part of the reason for this is because, as I said before to you, this is coming from metagenomic data. So we're trying to build some sort of pathway of recognizing functionality directly from metagenomic data without having to assemble these genomes and genes. And that's a different story, but we're looking at genes of these particular protein, training a Siamese neural network and asking whether two genes encode proteins that have the same function. And what we found is that indeed, this method works to recapitulate the functions that we have, right? So it mirrors the sequence similarity-based annotations, but it also does something weird. So I'll give you an example. Remember those fusion clusters, fusion functions that we had? So it was very high precision. So within one fusion function, you will have only proteins of one EC number, enzyme commission number, for example, right? But enzyme commission numbers would be distributed across fusion clusters. So multiple fusion clusters could have the same enzymes, and because the recall is fairly low. And so what this method does, even though it was not trained to do that, is that it recognizes that enzymes are actually similar functionally, even if I'm telling you that they come from different functions, right? So what you're seeing here is the scores. The y-axis is the prediction score from the Siamese neural network. And the data that we're running is comparing enzymes with a particular EC number that happen to reside in one fusion functional cluster. That's the big fat green bar on the left. And then we're comparing the same EC number versus different EC number, sorry, on the right. And we're comparing the same EC number versus different EC number in that brown versus green. And what you're seeing is that proteins that come from different fusion functions and also have different EC number score significantly lower than proteins that come from different fusion functions and have the same EC number. So this method, even though it was not trained to do that, had learned to recapitulate functionality in this particular space. And what we also observed is that if we split the space by structural similarity, right? So if we looked at proteins that have structures, aligned the structures using fold-seq and got the TM scores out of there, what we see is that, I'm not going to right or left, I guess that's right, but the part that says 0.7 to 1, so this is the highest structural alignments, actually in that space, the SNN differentiates proteins as being of the same function much better, right? So even if proteins share a structure, which is that brown orangish bar on 0.7 to 1 range, right? They score lower in SNN than the ones that share function, right? So this is an interesting observation. It captures structure to a certain extent. It was never trained to do that, but it does it anyway. Okay, so what we thought of is that if we were using sequence alone, which is the bottom curve here, the yellow curve, the bottom yellow curve here, is the performance and precision and recall space of our SNN method. It's actually pretty bad, right? It doesn't do well in recognizing sequence shared functionality. But if we add structure to it, which is the dashed lines on top, all of a sudden they perform really well. Actually, even better than just using structure alone, right? So we thought that the signal that we're getting out of this model is actually orthogonal to structure evaluations, and we can use the two together in order to evaluate functional similarity of organisms. So what does this give us, right? So all of a sudden, we can have a data set of proteins for which we have no idea what the function is, but we know that they share function, and we can actually ask how well do the methods that annotate functionality perform. And the reason, the motivation for this is whether deep learning models actually speak the language of life. We want to know whether that's happening for functional space. And again, the Pokeball, we want to know whether our tools will work for this space of unknowns, right? So we went out there and we got proteins to evaluate. So there are, at this point, 722, but it used to be 650 million, that's the second from the top bar here, 650 million that are representative of 2.5, I don't know what happened to that number, but billion sequences in Magnify. Magnify is a repository of metagenomic data. There's billions of sequences there that nobody knows what they're doing. They actually cluster into 600 million proteins. We looked at those 600 million and we said, for which of these proteins can ESM, which is this wonderful model that doesn't need multiple sequence alignment to predict structure, for which of these can we predict structure? And high reliability structures were only available for about 1% of the data, right? So my statement number one is that if we're going to look at proteins that have structures, we're looking at about 1% of the data. That means we're throwing out all the proteins that don't have structures, right? So disordered proteins that could have functions that we're interested in. We seriously can't evaluate them, right? So we looked at that 1% of the data, and then we asked, okay, well, how many of these are new? And in reality, there aren't that many that are new, right? That little sliver over there of 50,000 proteins or so is the new proteins. And what I'm trying to tell you is that everything I talk about is really not about things that are truly unknown. They're sequence similar, they're structure predictable. So we're just going to focus on these subset of proteins that is not the truly new ones, right? But somehow describable using the techniques that we currently have. Out of these 50,000, we selected proteins of length more than 400. Proteins translated from truncated contigs, that's the word contig over there, were removed. And then redundant proteins were also removed. So we actually reduced the redundancy of this data set to only the proteins that are less than 90% sequence similar. And we got 11,000 proteins out of this space. And 11,000 proteins, if you consider how many pairs that is, that's about 65 million pairs that you need to consider. But the good news about it is that protein structures did not align for most of them, right? So for protein structures, we have 305,000 protein structures. This is how they distribute across the TM score and the SNN score. So again, we are interested in that top space over there. This is the really well predicted, the very reliable pairs of proteins. So each dot is a pair of proteins. And what we're interested in is how well can other methods predict those on top over there, right? That right corner. There were, I don't know what's going on. There were only 6,000 proteins. I'm going to go on, but there were only 6,000 proteins in that set that were actually functionally similar. So 6,000 out of 305,000 were annotated to be functionally similar. And so how are we going to evaluate how the methods do? Well, For each pair of proteins, we're going to make predictions. So for example, method 1, that's the 2 predictions for protein, pink protein and a green protein. And we're going to compute a Jacquard index. We're going to compute the Jacquard similarity of the protein function labels that are produced by method 1 and by method 2. And what we see here is that the Jacquard similarity of the first one is 0.75 because we've got 6 out of 8 overlapping labels in this particular case. And then for the second one, it's 0.57, right? And we can do this for proteins that we labeled as siblings, so functionally similar proteins, and for proteins otherwise. And if we do that, we'll see, hopefully, that for dissimilar proteins, we get a worse performance, that similarity is lower. We measured the performance of the method as the delta S, the difference between the similarities of proteins that we knew were siblings and the proteins that were not siblings, right? So basically, your idea is that the higher the delta S, the better your performance is. Okay. So some methods don't just predict the actual yes or no. This is the label of the function. They will give you a score of the function, right? And for those of you who are users of these methods, the problem is that you need a score in order to know what is the actual prediction. So we computed the highest score by varying the thresholds for prediction. So for example, at a threshold of 5, we have some predictions that disappear, right? And we can compute the delta S in this case as well. So for all methods, we computed the highest difference between similar and dissimilar pairs. So if that was too fast, all you need to know is that the higher delta S indicates a better difference, better ability of a method to predict proteins that are of the same function versus proteins of different function. Okay. And if we look at the space of existing methods, so this is just some of the methods, you'll see that they map their protein pairs in all kinds of different spaces. So if this is all data, the small thing on the left, these are the pairs of proteins that each of these methods identifies as being functionally similar. And you could see that there is a distribution of dots in these graphs that is not equivalent. So these methods are orthogonal in terms of what they predict to be proteins of the same function. Curiously, all function predictions capture structure. So if you look at the same sort of interpretation of the data across the TM scores, so if you're closer to a higher TM scores, the structures are more similar. Everybody gets better as you get closer to structurally similar proteins. So structure does inform function, right? But some methods actually also are above random at every space of that particular curve. So random is the black line here, and the colored lines that are above random at scores of TM scores below 0.7 are the ones that don't actually rely necessarily on protein structure directly, right? And of course, this is the image that I normally look for. How do the methods perform specifically? So what you see here is that within each group of annotations, right? So we have orthology-based annotation, PFAM annotations, Go annotations, EC annotations, right? There is a leader. There is a method that performs better than others at differentiating same function versus different function proteins. And I'd like you to focus on PFAM for a bit. PFAM is not function per se, right? So this is basically understanding the domains of the proteins. And ProteinN is this deep learning-based prediction of PFAM domains. And what we see very clearly from this evaluation, for example, is that Hammer, the original version, the original Markov models that annotate PFAM, do better than the deep learning-based novel method for predicting PFAM domains, right? That's one thing. And the other thing where we see a lot of deep learning is Go-term prediction. And for Go-term predictions, it seems that it actually doesn't do very well, with the exception of the one method that's GoPredSim up there. And I'm telling you, I'm not biased. This is just the performance. All right. So if we wanted to do something that you guys are more familiar with, even though I don't like this whole scanning across the thresholds for predictions, you see that if you use the F1 max measure, no method performs well. If you use the area under the curve measure, then some methods perform better than others. And most methods do better as you measure the area under the curve, right? But again, as a user, you cannot use the area under the curve. You need to have an actual prediction of function, right? Okay, so here's the money shot. This is all the wonderful deep learning models that do not do nothing at all in identifying protein function, right? So that y-axis over there is to 0.05, but it really isn't. The best performing method here is Looking Glass, which is our method. And the reason, I will tell you the reason why it's the best performing, because the embeddings of Looking Glass is what we used in order to train SNM. All right. Okay, so the final result is that nobody does well. I think that's the conclusion in this case. So what do we need in order to improve our function prediction? We need to develop a forward-looking ontology for the yet unknown space. So these are the functions. Think of it as a Mendeleev table, right? Where we didn't know some of the elements, but we could say what they were. If we could predict, if we can create a function ontology that tells us what is it that we're missing in biological annotations, that would be very useful. I don't know how to do it, so if you have any ideas, that'd be great. And then the other thing is we can focus on the exploration of unknown functionality of known proteins. And I think that has been not really well happening at this space. So this is known proteins, unknown functionality, and unknown proteins, unknown functionality. In the generative space of the world, unknown proteins is something that we can actually think about. Okay, how much time do I have?
You're over by 1 minute.
Oh. Okay, I'm going to skip all the talk about metagenomes, and I'm just going to talk a little bit about this whole idea. I loved DALL-E when it came out, right? And basically, the idea is we take a submarine and you take a cat, and if you ask a method to predict other submarines or other cats, the method will probably do fairly well, right? So this is what we've been doing. We're taking proteins in the known space and predicting functionality in the known space. Here's the problem that I'm raising here, all right? So we now have this God knows what, and the current models will not annotate unknown functionality or proteins from this unknown space unless we believe that our models learn the language of life and it doesn't matter what are the texts that we have as long as we understand the language. So this is neither a cat nor a submarine, and neither of those models that I talked about before would be able to do anything good in this particular case, right? And they may be useful, but someone needs to evaluate that. So that's my message for this. This work was done by all of these people at Rutgers. I was at Rutgers up until very recently, funded by this money people, whatever. And I am now at this beautiful space. So I'm in Atlanta, Georgia at Emory. I am very happy there, but this is what my office space looks like. So if you're looking for a position, please let me know. Thank you.
Thank you so much. We have a few minutes for a couple of questions.
I want to go back to what you mentioned in the beginning. You mentioned that if you're working on proteins that we don't know the function, tough luck, and you mentioned kinases. But if you speak to some people in the kinase field, there are 300 out of the 500 human kinases that we don't know what the function. So it depends who you speak to. Knowing that it's a kinase is not enough. And for drug design, for example, or association with disease. And so I think we have to be a lot more precise of what would it mean by function. And it gets worse. I think it's a scaling problem. At every scale, it's the same problem of lack of precision and lack of annotation.
And the question?
How 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 can you make it not so superficial? Just to say that it's a kinase is not enough. It's not useful for the biologists. We like here we like to pat ourselves in the shoulder and say that we're predicting function, but you go to a biologist in an experimental lab and it doesn't help them. So how to make it actually useful?
So I think that you need to be making it useful at every point of your life, right? So that's why I didn't like this whole idea of scores, of thresholds, because you need to make a prediction and say this is blah, right? This particular type, right? The issue is that we are not there yet. It would be nice to have it. And this type of evaluation would allow you to do whatever you want at whatever level, right? So we need to figure out how to do that. We don't know how to do this yet. The reason I put up these slides is actually, I wanted to show you this. And so those of you who stayed, I'm going to talk about it for 2 seconds, if you don't mind. The idea is here, this is predicting oxidoreductases. This is a very high level, right? So EC number 1, just oxidoreductases from reads in microbiome data. And if we use any alignment-based method, so mgrest or myphaser, which uses the information that we currently have about what are the proteins that do oxidoreductase activity, we can annotate at most 40% of the data. That's mgrest right there. But with deep learning models, we can annotate a whole bunch of stuff, right? And now we have to evaluate how correct that annotation is. And we need these experimental biologists that you mentioned to actually do that, right? And we can't do everything ourselves. We need some interaction with people. And so what we did here is we validated it in terms of where the location of the metagenome is. And the expectation is that the oxygen minimum zones are supposed to have significantly more oxidoreductases than the surface area, which is the other side, right? Simply because in the absence of oxygen, oxidoreductases get very diverse. So the only method that was able to recapitulate that trend is the one that, the LSTM-based one. So none of the function annotation that we do with homology modeling is actually helpful in doing that. Right. So we're getting these wrong trends. Not only are we not getting what the biologists want, we're also getting the wrong trends by looking at homology-based annotations. Right. So the whole idea here for me is to, yeah, I agree, we need more precise annotations. But we should even start from the beginning, right? From this high level oxidoreductase space. Yeah.
Why do you specifically look at DNA sequence and not amino acids for your predictions? And sorry, I didn't answer in the microphone. Why do you look at DNA sequence and not amino acids for your predictions? And as a follow-up, the DNA sequence might move with the organism. So like thinking about codon usage bias, whereas the amino acids tend to be more related to function. So do you think you might be losing precision on that?
It's a very good question, or two questions. So to answer the first question, it's because I want to look here. It's because I want to look at reads, not at complete genes, right? So I'm still hopeful that I can identify functionality encoded in genomes or in genes on the basis of reads, right? And that will happen. But the other point about the horizontal gene transfer of genes across organisms, I think that the environment selects not just the codon bias in organism, but also the functionality. So what we're seeing actually by looking at the read embeddings is that environment is actually a big driver of what the reads look like. And I think that actually adds information, not removes information as compared to amino acids. Yeah.